Hey friends, welcome back. This is B Monica Sharma and welcome to MS BioAcademy. And as I have promised, I'm going to start from today onward 100 multiple choice questions discussion here in this videos. Okay, so there will be a series of videos for this. In every video, I'm going to cover up some 10 questions. I'm going to discuss it, and we are going to not only just going to discuss the question and answers, but we are going to discuss in between the details about some of the questions if it is required. Right, so let's begin it. So the first question is the longest gene in the human. So let me tell you again that the questions which I am discussing to you, all these 100 questions which I'll be starting up in this series of videos, these are the questions which are important for your exam point of view, and they have already come in the previous years in most of the competitive examination. So that's why I've taken all these selected questions, and I think you should know about everything. So this is a little bit of a preparation for you, a quick preparation for you, rather than just searching out on the net or on the books and reading about it. This will be the easy way to prepare for your future exams, right? So let's begin it. So the longest gene in the humans is what? The options are insulin gene, dystrophin gene, oncogene, and the beta globin gene. So this is very unique thing. You should always remember that the one of the longest gene in humans, in our body, is of the dystrophin. Dystrophin is a muscular protein, okay, and it is being coded by the DMD gene, and this gene is very long. This is about some 2.3 megabases, and this is present at a chromosome number 21, that too on the P arm. You know that the chromosomes have the two arms, right? Short arm and the long arm. So it is the P and the Q arm. So it is present somewhere in the P arm of the chromosome number 21, and the location is basically on the, you can say like this way, that it is P21, XP21. This is the location of the this particular dystrophin gene, which is coding for the dystrophin protein, which is a muscular protein. Okay, and this is one of the largest protein in our body. Okay, muscle protein. This is the largest muscle protein, you can say. The largest muscle protein is the dystrophin. So these are the important points you should remember, that the largest muscle protein is which one? Dystrophin. The longest gene in our body is which one? Dystrophin gene. Okay, so DMD gene or dystrophin gene. DMD word comes from because the mutation leads to the formation to the disease name as Duchenne muscular dystrophy, right? So that's why the DMD has been come. Okay, so let's move on to the next question. Question number 2 is cDNA, okay, is copied from the mRNA molecule with the help of. So cDNA, if you remember, cDNA is copied from the mRNA. So the normal central dogma, we know that what is the normal central dogma, says that from DNA RNA copy is made by a process known as transcription, right? Then from an RNA by a process known as translation, what it forms? Yes, protein, right? So this is the normal central dogma, which it says that always the information flows from DNA to RNA to protein. But sometimes what happen is there is a one opposite of the central dogma also happens, where at the level of this position, where the DNA copy is being formed from the RNA. Okay, so this is known as a reverse transcription. Since this process from DNA to RNA is known as what? Transcription. This is known as reverse transcription because we are reversing it from RNA to DNA, and the enzyme involved here is the reverse transcriptase. Okay, since the name from the reverse transcription, the name of an enzyme is reverse transcriptase, which is responsible for the formation of a DNA copy from an RNA, and this DNA which is now formed from the RNA is known as a cDNA. Okay, so here the question is cDNA is copied from mRNA. That means we are talking about the reverse transcription here from mRNA molecule with the help of. Now easily you can answer this question. Yes, it is reverse transcriptase. Okay, so this is the answer. Next is the tRNA has a unequal free arms. The option is short arm ending in the G and the long arm in CCA, short arm ending in A and long arm in ACC, short arm ending in G and long arm in CGA, short arm ending in C and long arm ending in GCA. So for this you need to remember the structure of the tRNA. In the tRNA you will see here that this is a clover leaf like structure of a tRNA, right? And at this position you can see that this is the long arm and this is the short arm, right? So you can see here this is a long arm, this is a short arm of the tRNA structure, and at the long arm you can see the sequence is CCA, while in the short arm it is the G, right? So now look at the option. So it is the short arm in G and long arm is CCA. So this should be the option number 1 is the correct, right? Otherwise there is one more with a short arm G, but the long arm they are saying it's CGA, and it's not CGA here. If you can see here, its long arm is ACC, right? Or CCA you can say, right? CCA. So first option is the correct for this. So this you have to remember. Okay, this you have to remember. These are the anticodon, right? If you remember these codes with the mRNA, complementary mRNA position, so they pick up as according to the whatever the information is there in the mRNA, they will pick up like that the one amino acids corresponding, right? So that's how the information flows. Now question number 4 is the DNA gyrase is a type of. So DNA gyrase, if you know, this is an enzyme which is basically a topoisomerase enzyme. So topoisomerase is basically an enzyme which is responsible for the DNA topology, means whenever there is a DNA replication take place. So if you know that normally not in the linear DNA but in the circular DNA, what happens is normally the CCC, the complementary covalently closed DNA, what happens is whenever the replication take place, so due to its double helical structure, what will happen, DNA will not be able to open it until and unless it needs to be cut from one side, right? So the replication fork will be very difficult to make. Just imagine it, if the replication fork is making, what will happen, just imagine a telephone coil. Okay, so if you are going to just unwind it or you need to just open it, what will happen, behind part will become more coiled. Okay, the moment you will start opening this thing, so the behind part will become more coiled or super coiled. So the same thing happened here. So what happened is normally the enzyme in order to avoid the super coiling or in order to maintain the topology of a DNA, what DNA gyrase is, it will cut one of the strand of a DNA, and then from one of the strand of a DNA, let's suppose it is cut. So now what happen is this portion of the DNA can come over from above the other strand, okay, and again then join in. So what will happen, it will remove that super coiling thing which is happening and again relax the DNA, right? So this is actually a name called as a twist, and there is a rith happens, okay, and there is a linking number. So what are all these things, if you comment it down below in the section, I can take a separate video on that, okay, and I can explain it to you what all these things means. This is related to a DNA topology, okay. So if you'll understand the DNA topology and all these meanings, then you will be able to understand the actually what the DNA gyrase is actually doing, how it is uncoiling it and why it is important, right? So here just remember that DNA gyrase is a type of a DNA topoisomerase. Now fifth question is, nuclear DNA sends the information for the protein synthesis through mRNA, tRNA, rRNA, or all of them out. Nuclear DNA, okay, sends the information for protein synthesis through what? It sends the information for protein synthesis through mRNA, tRNA, rRNA, or all of the above. It sends through the mRNA. tRNA it doesn't sense. tRNA just come at the later point. They read the mRNA information and pick up the amino acids and try to add up in order to synthesize the protein. But they are directly not involved in the information to be sent from the DNA to RNA. Again the central dogma comes, DNA to RNA to protein is happening, okay. So who is passing the nuclear DNA information is passing, who is passing for the protein synthesis? It is the mRNA. mRNA is passing it, right? So the option is mRNA. Don't take all of the above here. It is the mRNA. Question number 6 is that adenine is 30%. What would be the percentage of guanine? So if you know the Chargas rule, it says that according to the Chargas rule, it says that adenine is equals to thymine and amount of guanine is always equal to the cytosine. So here it is said that adenine is 30%. So that means thymine is also 30. Okay. Thymine is also 30%. So the total adenine and thymine will be 60%, right? And rest of them will be obviously guanine and cytosine out of 100. So it will be guanine and cytosine should be 40%, right? So guanine plus cytosine should be 40%, and we are saying that guanine is equal to cytosine. So we can write instead of cytosine guanine only here. So it will be 2G is equals to 40%, or you can say G is equals to 20%. So the answer for this is guanine will be how much? 20%. So this is very easy way. You can just take it out for everyone. Let's suppose if something else is mentioned here instead of adenine, you can just use this thing and find out the percentage of the another base. Okay. 7th is information from the DNA was conveyed by. Again, this is the same question which we have discussed earlier also. The information from DNA was conveyed by the mRNA. Again, the central dogma thing is coming here. Okay. So do remember, it is the mRNA which is conveying the information from the DNA. Now, 8th question is assertion. It says that HnRNA is larger than the mRNA. You know HnRNA is the heterogeneous RNA. Actually, HnRNA is the heterogeneous RNA which is basically a pre-mRNA. So what happens is from the DNA, whenever the transcription happens, so what we are saying, from the DNA, RNA is getting prepared by means of transcription process, right? But what happens is directly, since DNA contains lots of genes, so some non-coding genes are also present. That is, information is also being transferred to the RNA, right? So the first RNA which is being synthesized is the pre-mRNA. Okay, so this is the pre-mRNA, or you can say this as the heterogeneous nuclear RNA. Okay, so basically this heterogeneous nuclear RNA contains the non-coding portions also and the coding portions also, coding for the gene, for the proteins, right? So those are called as introns and exons. So introns are basically the non-coding portions. We don't need the introns portions, we need only the exon portion. So what we do is we do the process known as a splicing. So in which splicing this introns are being removed, and this heterogeneous RNA then form the mRNA which only contain the exons, the coding portions which are coding for a one particular protein, right? So this is how the information flows normally. So heterogeneous RNA is basically the RNA which contains the exons also and introns also, while the mRNA contain only the exons. The introns are removed after the splicing process. So now let's read the assertions. Assertion says that the heterogeneous nuclear RNA is larger than the mRNA. So yes, it is true, because HnRNA contains the exons also, introns also, but mRNA contains only exons. So obviously it is larger than mRNA. Reason क्या दे रखा है इसका? Reason is that HnRNA has non-translating introns, okay, and more exons than required for translation. So they are saying that HnRNA has a non-translating introns, which are the non-coding portions, introns are there, and more exons than required for translation. Okay, so it contains the more exons than required for translation. So this little bit is not convincing, that whether it contains the more exons than required for the translation. This is just a question mark, although till here it is correct. Okay, so now let's read it, the options. Both the assertions and the reasons are true, and reason is correct explanation for assertion. Okay, both assertion and reasons are true. Yes, both assertion and reasons are true, and the reason is not a correct explanation for assertion. I personally think that this should be the correct answer, and this is the correct answer for this, that both assertion and reasons are true, and the reason is not the correct explanation for assertion, because here it is mentioning that more exons than required for translation. Still I would I would ask you all if you can just comment in the section below your perspective of answers for this particular question, right? Because this is I suppose this should be the correct answer for this. Okay, so openly I would like to ask all of you if you can give the correct answer for this. Help the people, okay, help the students out there. So the next option is assertion is false. So assertion is not at all false, so this can't be the option. Assertion is true but reason is not correct. Okay, so this is also not completely correct, because reason is also half of it is correct. So this should be the correct option. Next is nitrogen base not found in a DNA is what? Adenine, guanine, cytosine, thymine or uracil? Obviously it is uracil. In DNA, instead of DNA, you will not find uracil. Uracil is found in RNA. RNA does not have thymine. Instead of thymine, they have a uracil, right? But DNA contain all these 4 bases. Nitrogen base not found in the DNA is uracil. Next question is gene does not produce what? Protein, sugar, polypeptide, enzyme, mRNA. Gene produce mRNA, gene produce enzyme, gene produce polypeptide, protein, but it does not produce sugar. So this is the option. Sugar is the option for this. So that's it for today. See you in the next lesson with some more questions. Bye for now.